Alanine is a glucogenic amino acid. It accounts for a large amount of the amino acids in most proteins. It can be synthesized from other amino acids, particularly in branched-chain amino acids. Alanine is released into the circulation in significant amounts during starvation and is easily converted in the liver by the catalytic action of glutamate pyruvate transaminase with ketoglutarate to produce glutamate and pyruvate. Thus, the pyruvate can be converted to glucose and be absorbed by the muscle to be used as energy source. Skeletal muscle produces and releases alanine, which the liver uses as a precursor for gluconeogenesis. It also can store energy as proteins and amino acids that are broken down during catabolic conditions like starvation. There are several functions of alanine, including play an important role in breaking off tryptophan and vitamin B6, provide energy to muscle and nervous system, as well as improve immune system in the body. There are several metabolic phases that involve metabolic pathways which are incorporated in protein structure. Formation of pyruvic acid, synthesis of glucose-alanine cycle, but in this video we are focusing on glucose-alanine cycle. Glucose-alanine cycle, also known as Khairil cycle or alanine cycle. This involves a series of reactions in which amino groups from the muscles are transported to the liver, and it is quite similar to the Khairil cycle. Alanine is transported to the liver and used for gluconeogenesis. Meanwhile, in muscle, pyruvate is converted to alanine, which in liver, alanine is converted to pyruvate. When this process occurs simultaneously, it is known as glucose-alanine cycle. This cycle occurs during anaerobic condition or increase in oxygen, such as extended period of fasting in which skeletal muscle is degraded as an alternative source of energy. Next, we will move on to the reaction that occurs in the muscle and the liver. First, let's take a look at the mechanism in skeletal muscle. In muscle cells and other peripheral tissues, glycolysis produces pyruvate. Then pyruvate can be transaminated into alanine. The transamination reaction requires an alpha amino acid as a donor of the amino group, generating an alpha keto acid in the process. This reaction is catalyzed by alanine transaminase. The alanine then enters the bloodstream and is transported to the liver. Afterwards, alanine is returned to the liver for gluconeogenesis. Then the mechanism in the liver, including alanine, is converted back to pyruvate by deamination. Pyruvate is a source of carbon atoms for gluconeogenesis. Gluconeogenesis converts pyruvate to form glucose. The newly formed glucose can then enter the blood for delivery back to the muscle. This pathway is termed the glucose-alanine cycle. This figure shows the difference between the Khairil cycle and glucose-alanine cycle. Basically, both Khairil and glucose-alanine cycle can depend on gluconeogenesis in the liver, followed by glucose output and liver to extrahepatic tissues. The only things that differentiate between them are the end products of glucose metabolism, in which the Khairil cycle produces lactate, while the alanine cycle produces alanine. Khairil and glucose-alanine cycles are important because they ensure the continued provision of substrate for gluconeogenesis. It should be emphasized that the Khairil cycle does not provide carbon for the net synthesis of glucose. Glucose formed from lactate merely recycles the carbon to the glucose to replace that which was converted into lactate by extrahepatic tissues. So that's all from us.